Hello, students. Have you ever wondered why scientists don't just stop at definitions in books? Why do they perform experiments again and again? That's because science is not just about memorizing facts, it's about proving them through careful observation. One of the best examples is germination, the amazing process by which a seed transforms into a seedling. Today, we'll use simple household materials like cotton, water, and jars to test the conditions needed for germination. Isn't it fascinating that everyday items can reveal the secrets of life itself? Now let's begin with water, the lifeline of all living things. Imagine placing seeds on wet cotton in one beaker and the same seeds on dry cotton in another. What happens? Seeds on wet cotton swell, soften, and sprout tiny roots, while the seeds on dry cotton just sit there, lifeless. This simple test proves that water is essential for germination. Here's why. Water enters the seed through a process called imbibition, softens the seed coat, and activates special enzymes inside the embryo. These enzymes break down stored food, giving energy for the radicle, the first root, to emerge. Without water, the embryo stays in deep sleep, unable to awaken into new life. Now, what about temperature? If you keep one beaker of soaked seeds in a warm room and another in a refrigerator, you'll notice a big difference. At room temperature, seeds germinate beautifully. But in the fridge, they remain dormant or grow very slowly. Why? Because enzymes, the biological catalysts inside seeds, only work in an optimum range around 25 to 35 degrees Celsius. That's why farmers wait for the right season to sow seeds. If the soil is too cold or too hot, germination just won't happen. Here's another hidden factor: oxygen. Suppose we place seeds in two flasks. In one flask, oxygen is removed using a chemical called alkaline pyrogallol. In the other, the seeds are left with normal air. What happens? The seeds with oxygen sprout normally. But the seeds in the flask without oxygen fail to grow. This shows us that seeds need oxygen for cellular respiration, the process that breaks down food and releases energy in the form of ATP. Without oxygen, the embryo suffocates and cannot develop into a seedling. Now let's bring it all together in the classic 3-bean seed experiment. 3 dry beans are tied on a glass slide and placed in a beaker of water. The top seed gets air but no water, the bottom seed gets water but little air, and the middle seed gets both air and water. Only the middle seed germinates fully. Isn't that amazing? This proves that both oxygen and water are equally necessary for germination. It's like 2 keys needed to open the same lock. Without one, the seed stays locked in dormancy. So, what have we learned? Germination depends on 3 essential conditions: water, oxygen, and the right temperature. Remove even one, and the seed will not grow. That's why seeds sown too deep in the soil often fail. They simply don't get enough oxygen. And this is also why agriculture, gardening, and even seed storage industries carefully manage these conditions. Isn't it powerful that such simple experiments, using nothing more than cotton, jars, and seeds, can uncover life's most important truths? I hope this journey into the experiments of germination has opened your eyes to how beautifully biology explains the miracle of life. Until next time, keep observing, keep questioning, and keep discovering the wonders of science.